discuss the structure of the skeletal muscle, and we will also discuss the ultrastructure of the skeletal muscle as well. Means that we'll discuss the muscle fiber and its internal structures as well. Now, first of all, let's suppose that this is a bone. Now, on this bone, let's suppose that this is a muscle. Now remember that muscle is always attached at two places or with two bones. Now let's suppose that this bone is humerus. Let's suppose this bone is humerus. This is the only funny bone in your body, humerus. This bone. Right? This is humerus. This one. Now, and this bone over here, this bone is called scapula. Now the bicep muscle is attached over here with the scapula and over here with the radius. Right? So bicep muscle is present over here. On the one side it is attached to the scapula and on the other side it is attached to the radius. Now this bone is called radius and this bone is called ulna, right? Ulna, U-L-N-A. So over here, this is the bone. So this bicep muscle, on one end, it is attached over here with this bone which is scapula, and over here it is attached with the radius. Understand?
Scapula is also called the shoulder blade, right?
Yes. Now, let me tell you, this scapula is an immovable bone. It's not moving, right? Rest of you see, my arm is moving, my scapula is not moving. The place where the muscle attach, where the bone is immovable, from that place, that is called as origin of muscle. The place or the bone where the muscle is attached and the bone is immovable, that side of the muscle is called as origin of the muscle. And the second place where the muscle is attached, where the bone is movable, like in this case, this bone, this is called as insertion of the muscle. So this is insertion, this is origin. Now muscles, they are usually tapering from both ends. I told you what is tapering when I was teaching you about the smooth muscle cells. They are pointy from both ends, right? So they are thinner from both ends, but from the middle, they are fatter. Now this middle, the fatter part of the muscle, this is called as belly of muscle. What is it called? Belly of muscle. Now, what I'm going to do, I am going to enlarge this diagram. Let's suppose that what do? I take a transverse section of the muscle. I cut the muscle from here and I observe it. Now what I will see? I will see the muscle like this. Now, sometimes if you see the muscle or when you are eating the meat, you feel that there is a layer around the muscle, there is a membrane around the muscle, right? Now that membrane which is around the muscle, I am representing it with this line, right? Showing that there is a membrane around it. That membrane is called as epimysium. What is it called? Epimysium. Now, in this epimysium is actually a connective tissue. It is holding the fascicles together. Now, question is in your mind, sir, what are fascicles? Now, I am going to tell you what are fascicles, but these structures that I am drawing right now, these are fascicles. Now, what are these? These are fascicles. Now, each fascicle is actually a bundle of muscle fibers. Now, if I enlarge one of these fascicles, let's suppose that I enlarge this fascicle, even around the fascicle there is what? A membrane. Even around the fascicle there is another membrane. The membrane which is around the fascicle, that is called as perimysium. What is it called? Perimysium. And I just told you that fascicle is actually what? It is a bundle of muscle fibers. Now, if you remember my last lecture, those of you who attended, that the skeletal muscle cells are also called as muscle fibers. Skeletal muscle cells are also called muscle fibers. So, if I am saying bundle of muscle fibers, it means that many muscle fibers, many skeletal muscle cells are present where? In the fascicle. Now, in one fascicle, there can be about 100 muscle fibers. There can be about 100 muscle fibers which are enclosed inside this. So let's suppose that these are what? These circles that I am drawing, these are muscle fibers or muscle cells which are enclosed by this membrane called perimysium. Now what do? If I enlarge one of these muscle fibers, now this is what? This is a muscle fiber. This is what? This is a muscle fiber. Now next step, what I am going to do, I am going to enlarge the diagram of the muscle fiber or a muscle cell, skeletal muscle cell I am talking about. Now, a skeletal muscle cell, you guys know that it is a syncytium. It is syncytium. Now this word is written in your book. Now let me tell you, any cell that has many nuclei or a multinucleated cell is called syncytium. A multinucleated cell is called syncytium. So if I say that the muscle fiber is a syncytium, it means that it has many nuclei. And which muscle cell? Skeletal muscle cell, we have learned that, right? Cardiac muscle cells are uninucleate. These smooth muscle cells, they are uninucleate. But only the muscle fibers, skeletal muscle cells, they are syncytium, they are multinucleate. Syncytium. Now, this is one of the muscle fibers. It's not whole muscle fiber. It's a part of the muscle fiber I have drawn over here. Understand? Now, first thing which is very important to observe, that these muscle fibers, they have special kind of channels in them, and these channels, they are called as T-tubules. So what is this? This is a T-tubule. Now what is the meaning of T-tubule? Transverse tubule. These tubules are arranged in the transverse way, right? So that's why they are called T tubules. In simple we say transverse tubules, simple way T tubules. Got it? With these T tubules, there is these organelles are present. Who will tell me what are these organelles? Excellent. Sarcoplasmic reticulum. Now remember that the endoplasmic reticulum like structures present in the muscles are called sarcoplasmic reticulum. So this over here, these are sarcoplasmic reticulum. Now let me tell you, this word sarco, it's a Greek word. Sarco is a Greek word, and this word sarco, it means literally muscle. It literally means what? Muscle. Then the word plasmic means cytoplasm. And the word reticulum, who will tell me its meanings? I have taught you last year. Not tubules, just the word reticulum means network. The word reticulum means network. Understand? So the complete meanings become inside the muscle cell, its cytoplasm, a network of channels. Network of channels. So this is called sarcoplasmic reticulum. Next thing that we discussed just right now, that these are what? Syncytium. There are many nuclei present. Now with the red color, I have represented what? A nucleus. Now as this is just one cell, I have drawn 3 nuclei to show that, to represent that this is a multinucleated cell. Next thing, we also discussed that these skeletal muscle cells, they are regularly striated. Remember that or not?
Yes.
They have regular stripes. What does that mean? It means that whenever we observe a skeletal muscle cell under the microscope, we see regular dark and light regions. We see regular dark and light regions. So this over here, this is the light region, and this over here is the dark region. Again, this over here is the light, and this one is what? Dark. The question is, why there are light and dark regions present in the muscle fibers or the skeletal muscle cells? They are present because these are actually representing sarcomeres. They represent what? Sarcomeres. Now question is, sir, what are sarcomeres? Again, these are 2 words, sarco and mere. Sarco means simply what? Muscle. Who will tell me what is the meaning of mere? You have learned that. Unit. These are structures present inside the muscle cell which actually help in contraction. What is the main job of the muscles? Contraction, right? So the structures that help in contraction, they are given the name sarcomeres, the units that help in muscles performing their job. Now, next step, what I am going to do, I am going to draw a sarcomere, right? And then we are going to learn its structure in detail. But still, one thing is left. Rather, few things are left. Now, I told you guys that around the whole muscle there is a membrane which is called epimysium. Inside the epimysium, the muscle cells are arranged in bundles which are called fascicles, and even the fascicles have membrane around them called perimysium, right? Now this is a muscle fiber or muscle cell. It has its own cell membrane. Now remember that it is a highly specialized cell. There are two types of cells that are very highly specialized, that their number never increase. Now one is nerve cells, if you guys remember I have taught you. The number of nerve cells increase up till the age of 2, I taught you that, right? After that, the degeneration starts. The number of these nerve cells starts decreasing. Second, the number of muscle cells increase up till puberty. After that, the number of muscle cells never increase. Then you will say, so those people who are bodybuilders, their muscles, they become bigger. The number of muscle cells do not increase. Size of muscle cells increase. Right? Now when the size of muscle cells increase, that is called as muscular hypertrophy. Let me just write over here. So that phenomenon in which muscle cells increase in size is called muscular hypertrophy. Now, till the time they are doing exercise, the size of muscle will remain larger. They will be in hypertrophy stage. If they stop exercising, muscle cells will come back to their original size. Right? Now, so it means that there is no increase in muscle number, muscle cells number. Now, as the membrane of the nerve cells, they had special names. What was the name? What is the name of the axon of its membrane? Exolemma. The cell membrane of the muscle fibers is called sarcolemma. What is it called? Sarcolemma. Understand?
And I thought you were practical.
What is there? That it is called sarcolemma. But another interesting fact. Sarcolemma is its cell membrane, but just like epimysium, perimysium, every muscle fiber has an extra membrane around it. Every muscle fiber has an extra membrane around it, and that extra membrane, just like you see, I showed over here and here. Here, I am going to show over here like this. That extra membrane present on the surface of the sarcolemma, that is called as endomysium. So there are 3 membranes overall total now. Epimysium around the whole muscle. Perimysium around the bundles of the muscle fibers. Fascicles, right? And endomysium around the muscle fiber itself. And remember, muscle fiber or muscle cell has its own cell membrane, which is called what? Sarcolemma. Next thing. In axon, its cytoplasm was called what? Exoplasm. Here, the muscle cell cytoplasm is called sarcoplasm. Yes.
Why is an extra layer? That was missing.
Very good question. Now, why there is endomysium? Now, you know that skeletal muscles, they are voluntary muscles. It's very important. Skeletal muscles are voluntary muscles. The message comes from the brain. Which nerves will bring the message from the brain to the muscle cells? Motor neurons, right? Motor neurons, they come and they spread on the endomysium. Motor neurons, their nerve endings, they come and they make synapse with the endomysium. Not only that, the endomysium, it also has blood capillaries. Every single muscle cell requires immense amount of energy to do its job, right? For that, there must be a proper supply of oxygen, a proper supply of glucose to the muscle cells, right? So that is only possible if every muscle cell has a contact with what? with the blood vessels. So the endomysium has what? Blood capillaries. Understand? So that there is easy supply of the glucose and oxygen to the muscle cells, and also you know that skeletal muscles, they not only perform aerobic respiration, they also perform what? Anaerobic respiration. When they perform anaerobic respiration, which chemical is produced in the muscle cells? Lactic acid. We know lactic acid is poisonous, right? So it must be removed from the cells. And you guys know that the muscle cells, they get rid of lactic acid, and lactic acid goes where? To the liver. Understand? For processing. So that also blood capillaries are required over there. So now you understand why the endomysium is required over there. Right? Now, question is, why the cytoplasm is given a special name sarcoplasm? Why was it given exoplasm? Because the cytoplasmic contents, they are different than a normal cell's cytoplasm. They have more ions in them. Remember, in the exoplasm there were sodium ions, potassium ions, calcium ions. Remember that or not? Over here the same thing is there. Right? Got it? Now, this was the overall view. Now we are going to learn what? Ultrastructure, and in the ultrastructure, we learn the structure of the sarcomere. We learn the structure of the sarcomere. Now, I am going to draw one sarcomere. Now, each sarcomere has, you see, these zigzag lines, right? Now, you draw these lines over here. Now, these zigzag lines, each zigzag line is called as a Z line. This is a Z line. This is a Z line. Now, in the middle over here, we have two types of filaments. We have learned about them in AS. Now, in this cytoskeleton, I have taught you, I am going to ask you questions from you. Now, these ones over here, this one is called a thin filament. This line with the black color I drew, these ones are called what? Thin filament. Then there are these filaments present over here. And this one over here, this is called as a thick filament. Let me label over here. What is it called? A thick filament. Now question is, why we are calling this one as thin filament and why we are calling it as thick filament? Because of the difference in their diameter. Thin filament has lesser diameter and thick filament has bigger diameter, larger diameter.
Yeah, so what is this again? What is this?
Whole thing.
The whole thing, what is it?
Sarcomere.
Just sarcomere.
This whole is a structure of a sarcomere. Now every sarcomere is in between two Z lines. I'm not finished yet. Every sarcomere is in between two Z lines, right? Next thing, that every sarcomere have two types of filaments. One is thin filament, other is thick filament, and their arrangement is like this. Their arrangement is like this. Next thing, now we are going to understand why there are these light and dark striations are there, right? When we observe this under the microscope, if you see this much area from here, Ibrahim, and this much area from here, of the sarcomere here, only thin filament is present. Can you see? This dotted line which I drew, in these areas over here and this area over here, only thin filament is present. It is thin. More light can easily pass through it. So under the light microscope, this area will appear lighter in color. This area will appear lighter in color. Now this area which is appearing lighter in color, this area is referred as I band. What is it called? I band. So this I band will be on the both sides. Alright? Next thing, if you see from here till here, from here till here, what is here? In between black and green line, this and this. Thin filament and thick filament are overlapping each other, right? Thin filament and thick filament, they are overlapping each other. As thin and thick filament are overlapping each other, less light will be able to pass through it. As the less light will be able to pass through it, observing under the light microscope, this area will appear darker in color. I did not say black, I said dark region. Right? So as this area will appear dark under the light microscope, this region is given the name as A band. What is it given the name? A band. So there is A band on this side, there is A band on this side. Right, Ibrahim? Please pay attention. So there is I band where there is only thin filaments. More light is passing. There is A band which is darker because both light, you see thin band and you see thin filament and thick filament, they are both overlapping each other. But if you see right in the middle, this area, only thick filaments are present, right? Only thick filaments are present right in the middle. Now this is thick filament. Can as much light can pass as it was passing through the thin filament? No. Light will pass less than thin filament, but more than the A band. Right? Now this region, this one I am showing it with the red. You see this bracket? This region is called actually as H zone. Now this H zone, it is darker than I band, lighter than A band. This H zone is darker than I band and lighter than A band. Why? Because only thick filament parts are present over here. Right? So the light will pass less than I band but more than A band. Understood? So that is why this is, you see, it is lighter than A band, darker than I band. This is called H zone. Next thing, right in the middle, I am drawing the line with the red color. So let me draw not the dotted line, proper line. Right in the middle, again it appears darker. Right in the middle of H zone or right in the middle of thick filament, it appears darker. Just a line of the dark appearance appears. This is called as an M line. The question is, why it appears darker right in the middle of the myosin filament or thin filament? I'm going to tell you the names of the thin filament and thick filament as well. Why it appears darker right? You see why this M line is there. Actually, this is a separate molecule and this is a separate molecule. When these two molecules they join in the middle, they give the appearance of darker appearance. Understand? This over here is a separate protein molecule. This is a separate protein molecules. They are bind from the middle. This is a separate protein molecule. This is a separate protein molecule. When they will come and bind in the middle, where the joint is there, it will appear darker. Not only that, right in the middle over here, there is creatine phosphatase enzyme is also present. Creatine phosphatase in the M line, right in the middle, where two myosin filaments, the thick filament, they are binding with each other. Now you do not need to memorize, this is just for your knowledge. Understand? You don't need to memorize it for your examination. Now, next thing, that this thin filament, it is also called as actin filament. It is also called actin filament. And this thick filament is also called as myosin filament. In AS, I have taught you the structure of actin filaments. Now, who will tell me which proteins are present in the actin filaments? Who remembers? Tubulin proteins were present in microtubules. Actin filament had three, I am making it easy for you, they had three types of proteins in them. One name is already written, actin. Troponin and tropomyosin. You learned that, right? That actin filament is made up of three types of proteins. Actin protein is already mentioned over there. Now here I have a question. This actin protein, which kind of protein is it? Globular protein. Actin protein is actually what? Globular protein. You guys have forgotten this. Right? Now, if let me tell you, if I draw the structure of an actin filament right now, I'm going to draw it for you guys. It's a reminder for you guys. You guys have forgotten. An actin filament now, this with the black line, this is tropomyosin I am representing. I am representing what? Tropomyosin. And with this red circles I am going to represent actin. Now, so these circles with the red color that I am drawing, these are actin proteins that I am drawing. Now, look here, with this black color, this black line, it is representing tropomyosin. These red circles, they are representing actin protein, which is a globular protein. Now, at intervals, troponin is present. Now, if you please pay attention, when I am drawing troponin, I am drawing what? Three dots together. So this blue one which I drew, this is what? This is troponin. I said that when I am drawing, now this is what? This is an actin filament that I just drew. There are three proteins. Tropomyosin is a fibrous protein. You can see its structure, right? actin is a globular protein, troponin is a globular protein. Troponin is present at different intervals, and troponin binds the tropomyosin on the surface of the actin proteins. Now, if you please pay attention, when I'm drawing, I drew this structure. I drew the tropomyosin outer to the actin protein. Can you see this or not? Can you see this or not? Right? Now this is an inactive form of the actin filament. This is the inactive form of the actin filament. Let me mention over here. Actin filament will be in this form when muscle contraction is not taking place. At that time, this actin proteins, they will be hidden. Their heads, they will not be exposed because they are covered by tropomyosin. Because they are covered by tropomyosin. Understand? And who is holding them? Troponin protein. And I just said that there are 3 dots of the troponin because troponin has 3 subunits: TNI, TNC, and TNT. Troponin has 3 subunits: TNI, TNC, and TNT. Understand? And it binds and it holds the tropomyosin over the heads of actin. Understood? Remember one thing, contraction will only take place when the heads of the actin protein will become exposed to the myosin. They'll be exposed to the myosin. Understood? After this, you see, this is the ultrastructure of the muscle. Next topic is the sliding filament theory, how the muscle contract actually. Understand? So, listen again, boys. Muscle, right? You understood about the origin and insertion of the muscle. Where it is attached to the immovable bone, that is called origin. Where it is attached to the movable bone, that is called insertion. The middle part of the muscle is called belly of the muscle. If we take a transverse section of the muscle, what we'll see that there is a membrane around the muscle called epimysium. Epimysium holds the bundles of the muscle fibers together. You see, and you see it has fascicles in it, which are actually what? Bundles of muscle fibers. If you observe a fascicle, now this is what? This is a fascicle. Fascicle also has a membrane around it called perimysium. In the perimysium, there are holding at least how much? 100 muscle fibers, right? Then what do we see? We increase the muscle fiber and I enlarge one muscle fiber, it's part of it. What did we observe? That there are T tubules, transverse tubules, right? With the transverse tubules, what is attached? Sarcoplasmic reticulum. Then they are multinucleated, we call it as syncytium. Its cytoplasm is called sarcoplasm and its membrane is called sarcolemma. Around the cell membrane, there is another membrane called endomysium. It has blood capillaries and nerve endings are present over there. Alright? Next thing, we discussed that in each, you see there are dark and light regions. Dark and light bands we can also say. We do say that, right? They actually make what? They make sarcomeres. This is one sarcomere. Each sarcomere has zigzag lines. In between 2 zigzag lines, there is one sarcomere. Right? Now, there can be another zigzag line over here, like this. So, what will be? This is another sarcomere. There can be another zigzag line over here, right? That will be another sarcomere. So, these sarcomeres, they are arranged along the whole length of the muscle fiber. Understand? There are thin filaments and thick filaments. Thin filament also called actin filaments. Thick filament also called myosin filaments. Why they are called thin? Why they are called thick? because there are difference in their diameters. Next thing, that the area which is only having the thin filaments that appears lighter under the light microscope, that is called as eye band. The area where both thin filament and thick filament are overlapping each other, less light will pass through it, that will appear darker in color, that is called as A band. The area where only thick filament is present, because it is thick, not thin, less light will pass as compared to the A band, but more than I band. So it will appear less darker to A, but more darker to I band. This is called H zone. And right in the middle, where two myosin filaments are binding together, joining together, that area appears as a one darker line, right? That is called as M line. Understood? So this is the structure of a sarcomere, right? And this is the structure of an actin filament. In the next class, inshallah, we'll continue with the structure of myosin filament and we'll learn the sliding filament theory, inshallah.
So this Z line is a chain of carbon atoms?
Not carbon atoms, this is a special protein over here. You don't need to understand which protein is this. You only need to know that this is Z protein. So Z line.
M line is practical or just animal?